Thank you, Alariza, for the introduction and the opportunity to come and speak to you today about my research into pharmaceuticals in soil plant systems. So I thought I'd start my presentation by providing a bit of background information on pharmaceuticals in the environment for anybody that isn't particularly familiar with this research topic. So thinking about pharmaceuticals in the environment, these can be both from human but also from veterinary use. And one of the most widely prescribed pharmaceuticals in human medicine, at least, is paracetamol. So I'd just like to get you to have a think about how much paracetamol as a UK population we take on a yearly basis. So I hope this will work, but in the chat function of this Teams call, if you could write down how much paracetamol you think on a per person basis we take in the UK, and this will hopefully frame some of the work that I'm going to present, or you can unmute your microphones if you want to come in with a suggestion. But hopefully I could get some responses from you and estimate how many paracetamols you think that you take, well, the British population take on a yearly basis. Is this in the tens of hundreds, a couple of paracetamol a year? Does anyone have any suggestions? Okay, so
No
we've got
idea.
one estimate so far, 20 to 40 per person per year. Okay. Is there any advance on that? Do we think it's higher or lower? Okay. Okay, so there's quite a variety here. So 20 tablets per year, 500 grams. So yeah, there's quite a variety of different responses. So in fact, it's been estimated that in the UK we take about 70 tablets per person per year when it comes to paracetamol. And if we gather that up as a total, then we're looking at about 6,000 tons of paracetamol that are sold on an annual basis. Now, when you take a pharmaceutical, you have a small portion of this that actually gets used in the body to elicit the therapeutic effect. And the pharmaceutical parent compound can actually undergo significant metabolism in the body. In the case of paracetamol, it's been estimated that about 95% of the drug can be metabolized or transformed, which leaves about 5% of the original pharmaceutical parent compound that's unchanged. And following the use in the body, this is excreted into the sewage system. So if we work on the basis that we're taking about 70 tablets of paracetamol a year, which is about 35 grams of paracetamol active ingredient, this will release in about 2 grams of the pharmaceutical parent compound through the sewage system on a per person basis. Now, if we work on the basis that we've got a population of about 67 million, we're looking at about 120,000 kilograms of active pharmaceutical ingredient entering our sewage systems. Now, to think about this in terms of mass, there's about the equivalent of 10 double-decker buses of pharmaceutical active ingredient for paracetamol entering our sewage systems. And what's important here is the example that I've just shown is just for paracetamol. So we have about 1,500 active pharmaceutical ingredients that are currently in use in the UK. And there's the potential for an unchanged fraction of all of these chemicals to enter our sewage treatment systems, which can then result in their release into the environment. So pharmaceuticals are now ubiquitous contaminants in environments. They've been detected in rivers and sediments and soils throughout the world. And the reason for this is that when these pharmaceutical active ingredients are entering the wastewater treatment plants, the treatment technologies are not specifically designed to remove or to degrade these chemicals. So they remain either in the effluent or they're absorbed to the sludge, which are then released into the environment. So hopefully this schematic will point out the key pathways by which pharmaceuticals can enter the environment. And my research predominantly focuses on the terrestrial or the soil compartment. So the pathways which I'm interested in are the use of wastewater effluent as a source of irrigation, and the use of sludge as a soil amendment or a fertilizer in their application to soils. But what we also need to remember is that pharmaceuticals are used in veterinary applications and for livestock, where you can get the release of active pharmaceutical products directly onto soils. And you can also get the presence of the antibiotics in the manures and the slurries, which are then used as a soil amendment as well. So there are a number of different ways in which the environment can become contaminated with pharmaceutical products. And it's really important that we evaluate this risk for a number of reasons. So with an increasing population, we're seeing an increase in use of medicines to sustain this aging population. And alongside this, we are seeing an increase in demand for agricultural produce. But there's also a drive to meet this demand for agricultural produce using sustainable agricultural practices. So this can include the use of alternative irrigation sources to reduce our reliance on freshwater irrigation, and also to reduce our reliance on the use of chemical-based fertilizers. And so we're looking for using sustainable agricultural practices, aligning with circular economy principles, and this is resulting in increased loadings of pharmaceuticals to soils. So it's important that we're able to establish what the risk is by introducing these compounds into the soil plant system. And this is important because these chemicals are bioactive in nature. As I pointed out in my first slide with the example with paracetamol, you have a proportion of the drug that is potent and retains this biological potency, and this doesn't disappear when it enters the environment. The potency of the chemical has, in fact, the potential to interact with receptors and non-target organisms in the environment. And it's important that we're able to establish the risk of this. And this is where my research really fits in. So today I'm going to talk to you primarily about my work looking at the uptake and the accumulation of pharmaceuticals in soil plant systems. And then I'm also going to touch briefly on some work where I've evaluated the risk to beehives through exposure in nectar and pollen, where the pharmaceutical residues have been able to be detected. And then lastly, I'm hoping to draw this all together and the key knowledge gaps which still remain as a result of this research, and highlight where I'm going to address those in my Future Leaders Fellowship. So starting with the work on the plant uptake and metabolism of pharmaceuticals, the first study I did with this was in around 2012, and this work was designed to look at how we can predict or explain the uptake of pharmaceuticals into plants based on knowledge about the fate of the pharmaceutical in the soil and properties about the chemical determining the uptake into the plant. So the three key findings from this study are presented here on this slide. So firstly, it's really important to know the bioavailable fraction of the chemical in the soil and soil pore water. And the bioavailable fraction can be influenced by sorption processes and also degradation. But it's important to know what's available for the plant to be taken up into the plant in the first place. So it's important to evaluate the fate of the chemical in the soil. You also need to have some understanding about the physical chemical properties of the pharmaceutical of interest. So some relationships that we were able to explore looked at hydrophobicity and then the accumulation in different parts of the plant. So more hydrophobic pharmaceuticals tended to preferentially absorb to the roots of plants, whereas if you've got a more lipophilic or a moderately hydrophobic chemical, then these can cross the root membrane but translocate into the above ground parts of the plant. However, if the chemical is really hydrophilic, then it stays within the soil pore water and isn't able to be taken up into the plant at all. So the distribution in the plant is a factor of the chemical properties. So if you have this information, you can start to build a picture as to whether pharmaceuticals are going to accumulate in plants, and if so, where they can accumulate, to start to think about risk. And then in 2018, I followed this work up with an additional study looking at the accumulation of a suite of pharmaceuticals, benzodiazepines, into plants. And what I did in this study was look at the accumulation in radish and the accumulation in two different soil types. So there was a sandy soil with a low organic carbon content, and then a clay soil with a high organic carbon content. And I was interested in looking at the differences in accumulation between these two soil types. And benzodiazepines were really interesting because I wanted to explore the potential for metabolism of these pharmaceuticals in the plants. So we know, based on human therapeutic pharmacokinetic data, that benzodiazepines can be extensively metabolized in humans, but we don't really know much about whether plants can metabolize pharmaceuticals. So this is where this study was designed, to try and explore that a bit further. So the initial findings suggested that the benzodiazepines accumulate to a greater extent in the sandy soil. And this links back to the slide where I talked about bioavailability. In the sandy soil, the benzodiazepines are going to be more bioavailable in the pore water for uptake into the plant in comparison to the clay soil. And we also saw a relationship between chemicals that were uncharged or in their neutral form, and those which retain some charge due to the pH of the soil, causing some ionization of the chemical. So the chemicals that were unionized, for example, oxazepam, were found to be taken up to a greater extent than the chemicals that were charged, such as flurazepam. But these were just some of the initial findings, and what I wanted this study to really do was explore the potential for metabolism. And so we're able to establish that the parent compound, as you can see in this example on the slide, diazepam, was taken up into the plant. But then I also analysed the plant material for potential metabolites. And benzodiazepines are really interesting because the metabolites of some of the benzodiazepines are actually prescribed as parent compounds in their own right. So we know, based on human data, that diazepam can be transformed into temazepam, which is also used in the treatment of anxiety. And we were able to detect the presence of metabolites in the plant material that was harvested. So this slide is just showing, on the left-hand side, accumulation of the parent compound diazepam, but also the concentrations of metabolites in both the sandy soil on the left side and soil with a high organic content and clay content on the right-hand side. And what I'd like to draw your attention to is the bars in the blue ovals. So this is the concentrations of diazepam in the soil and the plant material. But you can also see we've been able to detect concentrations of temazepam, nordiazepam, and oxazepam. And we're seeing higher concentrations of these metabolites in the sandy soil than in the clay soil. But they follow similar patterns to what we would expect based on the human metabolic pathway. So if you take diazepam as a parent compound, the first stage of metabolism is into nordiazepam, and this is through the loss of a methyl group. And this is a really fast stage of metabolism in humans. And as you can see here, the concentrations of nordiazepam in the plant are really quite high, whereas the second stage of the diazepam metabolism is quite slow. And this is the conversion to oxazepam. And we are seeing concentrations of oxazepam in the sandy soil exposure, but these are at much lower concentrations. And we weren't able to actually detect oxazepam in the clay soil. So it seems like we're seeing similar patterns in the plants to what we would observe in humans in terms of metabolism. What's important to point out here is that this is in-plant metabolism. So in the soil, we were able to detect concentrations of the parent compound, but not the metabolites. We were able to only detect the metabolites in the plant material. So this is indicative of in-plant metabolism, because if it was through degradation or biological processes in the soil creating the metabolites, which were then taken up into the plant. You would expect to see the presence of metabolites in the soil, but this is what we didn't see. So this is just another example to point out that it wasn't just the diazepam that we saw metabolites, we also saw it for a range of the benzodiazepines that we studied. So chlordiazepam is another example, which there's a similar pattern occurring here. You've got a fast transformation into the primary metabolite nordiazepam, and then a slower stage of metabolism into the secondary metabolite oxazepam. But again, this is in-plant metabolism rather than metabolism in the soil and creation of metabolites which are then taken up into the plant. I'll just go back because the important finding from this study is that the presence of metabolites is something that we don't often think about when we're considering the risk in terms of environmental exposure. We consider the presence of the parent compound and the fact that that might be biologically active. But this study has shown that metabolites can also be present in plant material, and these can be biologically active, and they can have a potency which is equal to or greater in some circumstances than that of the parent compound. So when we're thinking about risk and we're thinking about exposure, it's important both to consider the presence of the metabolite and the parent compound. Now, moving on slightly to some recent work that I've been doing, it was published last year actually, and it's trying to then think about the accumulation of pharmaceuticals in plants and the link to antimicrobial resistance. So hopefully most of you will be familiar with the concept of antimicrobial resistance, and this is briefly the ability of the microorganism to withstand the effects of an antibiotic. So ultimately, certain antibiotics cannot be used to treat infections, and this has the potential to lead to a global health crisis. Now, it's important to remember that resistance is a natural phenomenon, and bacteria are able to require resistance genes over time. But the problem is now that this resistance is spreading, and the environment has been implicated as playing a key role in this spread. So there are a number of different ways in which this can occur, especially in the soil plant system. So this could be either through the presence of antibiotics exerting selective pressure on the bacteria, leading to an increase in the prevalence of resistance, but also you could have the presence of resistance genes in the manures and in the wastewater that are then applied to land, and then there's the potential for these to be taken up directly into the plants. Now, a lot of the work that I've done with plants is focused on plants that can be eaten raw. So this has implications for human health effects and the transfer of resistance to the human population if we're consuming produce that contains these resistant genes. So this is where my work on the accumulation of antibiotics in particular links into research into antimicrobial resistance. And one example that I'm just going to introduce here was a study that I worked on with collaborators in China. And in China in particular, there's a real, at the moment, a real push to reduce their dependence on chemical-based fertilizers. So there's a lot of organic fertilizer substitution. And this study was derived to look at the impacts of this on the cultivation of rice. So unsurprisingly, when you applied the organic fertilizer to these soil rice systems, we were able to see an increase in the antibiotic resistance genes that were present in the rice. Interestingly, we also saw a reduction in grain yield, which suggests that there's a toxic effect occurring here from the presence of the antibiotics. And this is something that I'm going to move on to next. But for the moment, I just want to focus on this, the movement of resistance genes and the accumulation in the crop. Because in this study, we looked at the exposure both under current carbon dioxide concentrations and then future predicted concentrations of carbon dioxide. So the predicted concentrations we're talking about were predicted for 2100 and levels of around 570 parts per million. And the way we were able to do this was in the study fields, we had these rings effectively around the soil rice systems, and they pumped out extra carbon dioxide to increase the local carbon dioxide concentration above the rice plants. And this study was really interesting because we were able to see that the organic fertilizer substitution under current CO2 conditions, as I said, increased the presence of resistance genes in the plant. But when you evaluated the presence of the resistant genes under elevated CO2 concentrations, we actually saw a decrease in the abundance of the resistance genes. This was particularly evident for resistance genes to sulfonamide antibiotics and tetracycline antibiotics. So this just goes to show how complex the scenario is. We're currently working through the reasons why this may be, and there's some follow-on research to do here, because a lot of the effects that you consider when you're thinking about greenhouse gas emissions and climate change are very negative, and rightly so. But this research is showing that there's another side to the story. For some environmental exposures, there were significant decreases in the abundance of resistance genes. So this would, in theory, help stop the spread of resistance when you're considering scenarios where you've got elevated CO2 concentrations. So that's a few slides on my work looking at the accumulation and uptake of pharmaceuticals into plants. And this work that I've started with, really, the idea was to try and explore the uptake and accumulation in terms of evaluating the risks to human health by consuming the produce containing these contaminated residues. But what I have moved on to recently is to start to explore whether this accumulation can result in any potential toxic effects. And this is because of the bioactive nature of the pharmaceuticals that I've talked about. So is there the potential for the pharmaceuticals, when they're in the plants, to interact with receptors that could potentially elicit a toxic response? So some of the work which I've done to explore this is looked at the effects on biomass. And here's just one example. So this is with zucchini leaves and the pharmaceutical carbamazepine. And the pictures on this slide just show that at increasing carbamazepine concentrations, we're actually seeing an increasing formation of dead spots on the leaves and burnt leaf edges. And the most prominent effects were seen at concentrations in the soil of this drug around 10 milligrams per kilogram. Then, to follow up on this study, I did a trial with zucchini again, but looking at long-term effects. So this was a greenhouse study, so the zucchinis had to be hand pollinated, and we had different treatment concentrations and also controls. And the controls worked beautifully. The pollination resulted in a bountiful supply of zucchinis. I wish we could have eaten them, but we couldn't. But what we were able to see was that at the high concentrations in this study, the female flowers were unable to set fruit. So we pollinated them in exactly the same way that we had done with the controls. But post the pollination, over 2 weeks post-pollination, the flowers and the fruits actually shriveled up and died, as you can see on this picture. So this is showing that at high concentrations, the drugs are able to elicit this toxic effect that results in the lethality of the plant. Now, the concentrations, as I've mentioned, were high in terms of environmental relevance. And this is just a map of where wastewater is typically used for irrigation. And one of the most well-studied sites in terms of the accumulation of pharmaceuticals is in Mexico, so in the Mexiquito and the Tula Valley regions in Mexico. And the concentrations that we're seeing of pharmaceuticals in this area are around the nanogram to microgram per kilogram concentrations. And this is a lot lower than the concentrations that we were seeing in our lab experiments, where you were seeing this toxicity in terms of lethality. But then this got me thinking that if we're seeing concentrations eliciting a response in terms of this manifestation in this biomass response, then are there concentrations, lower concentrations, that are resulting also in a toxic effect, but what we would call sublethal toxicity? So not necessarily the death of the plant, but could be influencing the growth and the development of the plant. And this is where my research into sublethal toxicity started. So again, I used zucchini. So I should probably mention that this work, primarily looking at toxicity of plants, was carried out when I was at CSIRO in Australia. So apologies because I keep calling it zucchini rather than courgette. But this was an example of a market gardening practice where they use reclaimed wastewater for irrigation, and one of the crops of interest there was zucchini. So this is why there's been a lot of work on this from my perspective, because of the links that I had with Australia. So this study was designed to look at the accumulation of two different pharmaceuticals. So carbamazepine, I've mentioned, is an anti-epileptic drug. We have characterized it quite well in soil plant systems. We know it's taken up to a high extent. And then I also looked at verapamil, which is an antihypertensive and something that we know less about in terms of accumulation in soil plant systems. And so I looked at effects on biomass, but then also effects on chlorophyll, the nutritional content of the plants, and phytohormone concentrations. Really, just to put this research into context, the first results which we were able to get were the impact on the biomass. So the biomass results for carbamazepine are on the left and verapamil are on the right. So on the x-axis, you've got soil concentration increasing in the concentration as you go across, and then you've got biomass as a percentage of control on the y-axis. So what I'd like you to take home from this slide is that for the carbamazepine treatment, at concentrations greater than around 1 milligram per kilogram, we were seeing a significant effect on the biomass in the soil plant system. So this wasn't so unsurprising based on our previous research that has shown at high concentrations you do see this effect. But in the verapamil treatment, we didn't see the same effect on the biomass. If anything, in some circumstances, the pharmaceuticals actually enhance the growth of the plant. So if we can remember that carbamazepine concentrations greater than 1 milligram per kilogram had a substantial effect on the biomass, whereas verapamil we weren't seeing as greater effect, if anything at all. So the results which I'm going to focus on for the sublethal effects are phytohormones. And for those of you who are not familiar with phytohormones, I just included this slide here to hopefully illustrate why they're important in plant growth and development. So when a plant is under an abiotic or biotic stress, then this results in the complex network between the production of plant hormones and the pathways initiated by these external cues. And ultimately, this manifests in a stress response in the plant. So if we take a plant that's under water stress or drought conditions, for example, then the phytohormones are responsible for sending the signal for the plant to close its stomata to prevent any additional transpiration loss. And then the plant is able to restore its water balance. So phytohormones have an integral role in terms of plant functioning. So I analysed the plant material and looked for 4 different phytohormones, so cytokinins, auxins, abscisic acid, and jasminates. And the slide here presents the results for cytokinins and auxins. So again, you've got carbamazepine on the left and verapamil on the right, with the increasing soil concentration along the x-axis. So what I'd like you to draw your attention to are the bars which are highlighted in the circles. So for the carbamazepine treatment, we're actually seeing a higher concentration or an excess production of plant hormones at the lower concentration ranges for auxins and cytokinins. In the verapamil treatment, we're seeing this excess production and elevated concentrations of the plant hormones across all of the treatment concentrations. Now, these two specific plant hormones are responsible for cell differentiation and organ growth. So if I can draw your attention back to this slide here, where we're looking at the effects on biomass, we're seeing no effects on verapamil biomass and effects on the carbamazepine biomass at concentrations greater than 1 and 2 milligram per kilogram. Now, interestingly, this is where we're seeing the difference in the production of these plant hormones. We're seeing an excess production and higher concentrations of the plant hormones where we didn't see an effect on the plant biomass. So what we hypothesized is that this excess production of these plant hormones may be responsible for shoot growth stimulation. So the pharmaceutical in the plant is actually having an effect, but there's a compensatory mechanism for the effects induced by the pharmaceutical uptake through the production of these plant hormones. So where the plant isn't able to sustain the excess production of these plant hormones, here, where you're seeing a decrease in the concentration, this is where we're seeing an effect on the plant biomass. And then moving on to abscisic acid and jasminates. So these plant hormones are important signaling molecules that regulate plant defense responses. And what we were able to show is that at environmentally relevant concentrations, so here we're talking of very low microgram per kilogram concentrations, there's a significant difference in comparison to the control for the concentration of these hormones. And now this is really important because these are signaling molecules for plant defense responses, and this would suggest that at these concentrations, at environmentally relevant concentrations, there's implications for plant disease survival. So how well is a plant able to respond to an additional stress under pharmaceutical accumulation? And this is something which I'm looking into to exploring more. So thinking about pathogen attack in plants under drought conditions and pharmaceutical exposure. Are the plants able to respond to this stress if you're getting this reduction in the plant hormone concentrations? Okay, so that brings me to the end of my work, really talking about plant soil systems in general. And I'd like to just move on at the end of this talk to introduce some work that I've been doing looking at the accumulation of pharmaceuticals in beehives. So this study was designed to look at the accumulation of pharmaceuticals in nectar and pollen. So we're able to demonstrate that pharmaceuticals can be taken up into plants, but then move to the nectar and pollen. And this was the first time that this has been able to be shown. So the concentrations at this where this accumulation occurred were up to about 370 nanograms per mil in nectar and about 30 micrograms per gram in the pollen. So this experiment was purposefully done at relatively high soil concentrations because I wanted to be able to detect the carbamazepine in the nectar and the pollen of the flowering plant. But as you can see from the figures to the right of this slide, there was a nice relationship between accumulation in the nectar and pollen and increasing soil concentration. So then this led me to be able to consider an environmentally relevant exposure scenario. So again, thinking about Australia and market gardening practices there, we estimated that a typical concentration resulting from this wastewater irrigation would be about 20 nanograms per kilogram of carbamazepine in the soil, which based on our relationships between the accumulation of carbamazepine in the nectar and pollen and the soil concentration, we're able to predict how much carbamazepine would be in the nectar and pollen at this environmentally relevant soil concentration. So that's the first step in the analysis. I then collaborated with some environmental modelers to then look at the accumulation or potential accumulation in beehives from this contamination of the nectar and pollen. So this involves some landscape modeling, and it was parameterized for South Australian conditions. So we've got a 25-kilometre square landscape here, and it's a representative field for intensive horticultural production. And the red patches are the zucchini fields that are contaminated with carbamazepine. The green is oilseed rape, and then we've got trees in blue. So this is a representative scenario. And then we used both Beehive and Bee Scout models to look at the development of the honeybee colony and evaluate the transfer of the carbamazepine from the soil via the nectar and pollen into the beehives. So this was simulated across 3 years with different weather or climate conditions, and the scenario involved having 11 different 11 beehives located within this horticultural region with different site-specific foraging ranges. So I don't have time to go into all of the results from this, but I think this slide summarizes nicely the key findings. So we've got foragers on the x-axis, we've got the percentage of zucchini within the site-specific foraging range on the y-axis, and carbamazepine accumulation in the beehive on the z-axis. And what we've been able to show is that when you have a low percent of zucchini in your site-specific foraging range for the beehives, then you see low carbamazepine accumulation. And this makes sense because if you don't have the contaminated crop, then you're not going to see movement or accumulation into the beehives. Where you see the greatest accumulation, this is when you have both a combination of a high percent of zucchini in your fields and a high number of foragers. So this is where you're seeing accumulation in the beehive up to about 2,400 nanograms. So now the next step in terms of this analysis that I'd like to follow up on is looking at larval toxicity, because we've demonstrated that there's the potential route of exposure to a beehive, but then we need to understand what this means in terms of risk. So that's something that I'm hoping to explore in the future. So that brings me to the end of the presentation in terms of the work that I've done previously, and now I'd like to just move on to the work that I've got planned for the future. So I've just started a Future Leaders Fellowship, and this is designed to create this science platform that uses a variety of different approaches to evaluate the risk of emerging contaminants in agricultural systems. So this is predominantly going to focus on pharmaceuticals, but we'll also consider other contaminants such as plastics, personal care products, which also might exert a risk in the soil plant systems. So this is going to look at exploring inputs into the environment, effects on soil processes, so follow up on the work that I initiated with sublethal effects and looking at how this can impact on plant or crop productivity, and then think about how this risk changes in response to a changing climate. Now, this work involves collaborations with experts across the world. So I'm going to be working again with Australia, but some researchers in Israel and the US as well. And I'm going to be employing postdocs and research technicians and PhD students to work on this project. So there's the potential for collaboration in this research project. So if anybody is interested, then please do get in touch. So I'd just like to finish by presenting some of the key findings in terms of my research. So we've been able to establish that pharmaceuticals can be taken up from soil and that they can accumulate into plants. And this is a factor of both the physical chemical properties of the pharmaceutical, but also the soil properties. We've also been able to show that plants can metabolize pharmaceuticals, and this is important because we need to consider both the risk that arises from the parent compound, but also metabolites. And the issue of sublethal effects, so this is still relatively new. There's a lot of research that I'd like to do in terms of this area, but the initial findings suggest that environmentally relevant concentrations, there's impacts on plant growth and development. So we need to consider this at a larger scale and understand the impacts on crop productivity. And then lastly, the work that I presented on bees, this is relatively new, so there's a lot more research that needs to be done here to understand the risk from both a wider variety of pharmaceuticals that are present and we know are in soil plant systems, but also consider the risk to the bee population itself. So I'd just like to finish by thanking my collaborators, which have been involved in the projects that I've presented, in particular, Alistair at York and Rye at CSIRO. So thank you for listening, and welcome any questions if you have any.